Hello, I'm Dr. Azaz from MedicoVisual.com, back with another visual lecture on coronavirus. It is a part of coronavirus lecture series. In this lecture, we will talk about the transmission of coronavirus infection and then we will talk about its replicative cycle. So let's start. So as you know that coronavirus, it is a respiratory virus. So with each replicative cycle of coronavirus, a large number of particles of coronaviruses, these are released into alveolar sac of the patient. These are released into alveoli of the patient. These are now ready to infect other humans. So when this patient coughs or sneezes, what happens that the bronchoalveolar fluid, it gets mixed with these virus particles and aerosols may be formed. So what are aerosols? Aerosols literally means flying solutions. Aero mean air or related to air, and sols mean solutions. So these are literally flying solutions. Basically this is actually bronchoalveolar fluid in which virus particles are dissolved. So virus particles are solute which are dissolved, they solute the virus particles, these are dissolved into bronchoalveolar fluid. So these are the aerosols. So they may enter into respiratory system of another person, another normal person, and it may cause infection in that normal person. So that's how coronavirus gets transmitted from one person to another, mainly through aerosol or droplet infection. The other route is that due to these respiratory droplets or aerosols, they may fall onto towels and other inanimate objects. So when they fall on these other inanimate objects, they will cause what? They will contaminate those objects, and when another person will use these objects, well, for example, let's say some person will use these towels, he or she may get infected with this coronavirus disease, right? So these are the two main sources of infection. Of course, close contact, it may increase the chance of close contact with the patient of coronavirus disease. It may increase the chance of coronavirus infection. Then there is a limited evidence of transmission of coronavirus infection through fecal contamination. So this was about transmission. Now we will talk about replicative cycle. Let's suppose this is a coronavirus and inside it is the nucleocapsid, outside is the envelope, and spike proteins or S proteins are there. Now in our alveoli we have two types of cell. These squamous cells, these squamous type of cell or flattened cell, these are called type 1 alveolar epithelial cells or type 1 pneumocytes. The other cuboidal one, these are called type 2 pneumocytes. Now this coronavirus, it loves to infect type 2 pneumocyte because ACE2 receptors are abundantly present on the surface of they are embedded in the cell membrane of type 2 pneumocytes, and these coronavirus, their S proteins, they can bind easily and nicely with these ACE2 receptors. Basically ACE2 receptors are actually enzymes. These are transmembrane enzyme. We will discuss its function when we will talk about its pathogenesis. So I have enlarged their type 2 pneumocytes and now we will see what happens. So as this virus particle, its spike protein, it binds with this ACE2 receptor, what will happen that there will be some conformational change into viral particle such that this viral particle, it's the envelope of virus, it gets attached with plasma membrane, and the nucleocapsid will be pushed inside the cell. Nucleocapsid will come inside the cell. Inside the cell, the capsid will be dissolved, and what we will get is the genome of coronavirus. And as you know from the previous lecture, the genome of coronavirus, it is single stranded positive sense RNA, right? So if you don't know about this, please watch that part 1 of this lecture series. In that I have explained clearly that what is the structure of coronavirus and what is positive sense RNA and what is negative sense RNA, right? So now what we have got is that we have got the genomic RNA. We have got positive sense genomic RNA of coronavirus. Now, right now, at this stage, coronavirus, it is worried about making its copies. It is worried about survival of its species. So, because positive sense RNA, it can directly act as mRNA. That is, it can be directly transcribed into. It can be not transcribed. Sorry, it can be directly translated into proteins. So what happens that RNA of coronavirus, not the whole RNA is translated. A small part of its RNA will be translated, and a special protein will be formed. Basically, these are two proteins that are formed, and they combine to form a single enzyme. And what we call this happy enzyme, this enzyme is actually RNA dependent RNA polymerase. What this enzyme can do is that it can make copy of RNA. It can make copy of this RNA, right? So it can make copy of RNA by looking at another RNA. So it depends upon RNA. It depends upon RNA to make a polymer of RNA. So because it depends upon RNA to make a polymer of RNA, we call it RNA dependent RNA polymerase. So are you getting the point? So another simpler name is viral replicase, because it helps to replicate the genome of virus. It helps to replicate the genome of virus. So other name is viral replicase. And because it is also involved in transcription, it is also involved in synthesis of mRNA of the virus. So the third name of this very enzyme is viral transcriptase. So RNA dependent RNA polymerase, replicase, transcriptase, all these are the names of single enzyme, right? So what this happy enzyme will do is that it will induce, this is the endoplasmic reticulum, this is the endoplasmic reticulum of host. So this happy enzyme, this replicase, it will induce the formation of double membrane vesicle. It will induce formation of double membrane vesicle. What are double membrane vesicles? As the name indicate, basically these are just double membrane vesicles. So detail of this is not known, that how it induces the formation, what is the exact mechanism. All we know is that after formation of double membrane vesicle, it become this replicase, it become associated with these DMVs. It become associated with the DMVs, and whole this complex of DMV plus this replicase, it is called replication transcription complex. It is called replication transcription complex. And what is its function is, this function of RTC is that inside the lumen of this RTC, it is believed that inside this lumen of RTC, replication as well as transcription occur. So replication and transcription of genome of coronavirus, it occurs inside these DMVs. Are you getting the point? So now that this complex has been created, it will create a copy of genomic RNA. It will create a complementary copy of genomic RNA. So genomic RNA, the original genomic RNA was positive sense, and when its complementary copy has been made, when its complementary copy will be made, it will be not positive, it will be negative, right? It will be negative. It will be negative sense strand, right? Let's suppose this is the sequence of original genome, and if we have to make a complementary copy, rather than A, here U will become, and here rather than U, A will be here, C, U, and so on. So there will be in front, if there is a positive sense strand, in front of it negative sense strand will be formed, which would be its complementary copy, right? Now this is a problem. Now our enzyme, our replicase enzyme is very sad. It had done lots of hard work to create this complementary copy, which is quite useless right now. Why it is useless? Because it is negative sense strand. So can this negative sense strand be used as a genomic RNA for another viral particle? Of course not. We cannot push this negative strand into another viral particle and form a new virus out of it. Why? Because coronavirus genome is positive sense. So this cannot be used as genomic RNA. Also, it cannot be used to synthesize proteins. It cannot be used to synthesize protein. It cannot be used as messenger RNA. Right? So this is a problem. So now we have to convert, now rather this replicase, it has to convert this negative sense RNA back into positive sense. And how it will do so? Well, it's very simple. Just like when we create positive, when we create complementary copy of positive strand, it would be negative strand. And when we create complement copy of negative strand, it would be positive strand. It is very easy to understand. So it has created a copy from this template. So using this negative sense strand as a template, it will create a copy, and it will run again and again onto this to make multiple copies. So multiple copies of this RNA will be made, and these copies, these are full length genomic RNA, full length positive strand continuous genomic RNA, which can be and which will be used as the RNA of new virus particles, right? So now that it has created lots of copies of genomic RNA, now it has to worry about creating viral proteins. So now it has to create viral proteins. So our hard-working replicase still has to do a lot of work. But let me tell you something interesting here. Let's suppose this RNA, its nucleotide sequences, let's suppose this RNA, we are just assuming that it contains 100 nucleotides. There is 100 nucleotides in this RNA sequence, right? So from 20 to 100 gene of S protein might be from nucleotide number 20 to 100. This is, let's suppose, sequence of, this is, let's suppose, gene of S protein, and gene of E protein would be from nucleotide number 40 to 100, and M protein is from 60 to 100, and N protein is from 80 to 100. So why I am telling you this? You will be very clear about this within a few minutes. Just listen it very carefully. Now what will happen, that to create RNA for S protein, messenger RNA for S protein, this replicase, it will have to enter from nucleotide number 20. It will enter from nucleotide number 20 and exit from 100. For creating E protein, it will enter from nucleotide sequence number 40. For M it will enter from 60, and for finally for N it will enter from nucleotide sequence number 80. So there will be multiple origin of replication. These are called origin of replication. So there will be multiple origin of replication, and you can see that when it will make complementary copy of this is the RNA for S protein. This is right now not the messenger RNA. I will tell you why. It will run from here to here and it will form messenger RNA, not messenger RNA, RNA for S protein. Then for E protein from 40 to 100 will be E protein. Then for M protein, and finally for N protein. S protein is spike protein, E protein is envelope protein, M protein is membrane, and N protein is nucleocapsid protein, which are also capsomers, right? So what we have seen that it has created negative sense strands. It has created negative sense strands for protein synthesis. But again, negative sense strands, they cannot be used for protein synthesis. So now this enzyme will use these negative sense strands as template, and it will convert them. It will make complementary copy of them and convert them into positive sense RNA. Now here I would like to tell you about something called nested subgenomic RNAs. Now if you look carefully, the part of the part of RNA of N protein, it is nested inside M protein. It is nested inside sequence of M protein, and M protein, you can see it is nested inside E protein. Here you can see, and E protein, it is further nested inside, its sequence is further nested inside sequence of nucleotides of S protein. So N protein nested into M protein, which is further nested into E protein, which is further nested into S proteins. So these sequence of RNA which are nested into one another, these subgenomic RNAs, these are of course not genomic, these are subgenomic, right? These subgenomic RNA which are nested into each other, these are called nested subgenomic RNAs. And when they will be converted after making complementary copies, positive sense copies of this, these are now nested subgenomic mRNAs. So that's what they call it. That in books you see written like this, that coronaviruses, they form nested subgenomic mRNAs. Coronavirus form nested subgenomic mRNAs. Now you must be clear that what is these nested subgenomic mRNAs, and because of this property, taxonomically coronaviruses, these are arranged under order of nidovirus. These are arranged under order of nidovirus, right? So now that mRNA has been created and our enzyme is very happy because its work has been almost completely done, right? So what next? Now from mRNA of N protein or nucleocapsid protein, capsid proteins will be synthesized, and it will use these free ribosome that are present in cytosol. It will use free or cytosolic ribosome to synthesize nucleocapsid protein from this mRNA of nucleocapsid proteins, right? So this nucleocapsid protein is now synthesized. So this nucleocapsid protein, it will immediately find its beloved genomic RNA. It will find its beloved genomic RNA and it will associate with this genomic RNA to form what? To form nucleocapsid, right? So now nucleocapsid has been formed. Now what? So okay, so this thing has been done. Now what about this M protein, E protein and S protein? Here I would like to talk about something else as well. these are not only proteins that are synthesized by coronavirus. There are some non-structural proteins as well which are synthesized during this process. These are simply called NSPs, or non-structural proteins. I'm not going to discuss the details of non-structural proteins, so you can just skip. And by the way, to remember the order of these nested subgenomic mRNAs, you can remember it from the acronym SEMEN, right? SEMEN. Okay. So now N protein has been created. How about these M, E and S? How they will be created? Now they won't be using the free ribosome or cytosolic ribosome. Rather, they will use those ribosomes that are attached with rough endoplasmic reticulum, right? Why they do so? Because as they are being synthesized, as the M protein, E protein and S proteins are being synthesized, their polypeptide, they enter inside the lumen of endoplasmic reticulum, and here they are further processed. They undergo co-translational and post-translational modification. They undergo co-translational and post-translational modification, right? And then these newly synthesized proteins, these S proteins, M proteins and E proteins, these are anchored into membrane of endoplasmic reticulum of the host cell. These are anchored into membrane of endoplasmic reticulum of the host cell, right? So now what will happen? This nucleocapsid, it will go inside this endoplasmic reticulum, right? And then it will pinch off a part of endoplasmic reticulum where these proteins are present and where this nucleocapsid was present. It will bud off from endoplasmic reticulum, and here we have our beloved Golgi apparatus. Then this vesicle will go and fuse with Golgi apparatus, and then Golgi apparatus will pack them into special vesicle. They will think that this is something very useful. Golgi apparatus will think that the cell has created some beautiful proteins, so they will pack it into special gift packs. Golgi apparatus will pack them into special gift packs called vesicles. So these vesicles, they will be exported out of the cell by the process of exocytosis. So these deadly gifts will be exported out by exocytosis. And with each replicative cycle, here I have shown only a single viral particle being synthesized, but with each replicative cycle, thousands of particles are synthesized, right? Secondly, these spike proteins, these are also anchored onto plasma membrane of host cell, and which may lead to immunogenic response against these infected cells, right? So now that these viral particles are formed, they are now ready to infect other type 2 pneumocytes of the body, and they may also infect another person as well. They may infect other human as well when they will be coughed up or sneezed, as I have discussed earlier in transmission, right? So this was the replicative cycle. Okay, so are you clear about this? Okay, so next we will discuss pathogenesis, right?